Hello to all the listeners of Inside Matters. I'm pleased to introduce Dr. Debra Silberg. She is the Chief Scientific Officer of a wonderful charity called Beyond Celiac. And she's a highly accomplished life sciences executive with deep expertise across all stages and phases of drug development. Of course, medical doctor by training, but she's worked in industry and had clinical leadership roles at big companies that you'll have heard of before: Shire, Takeda, AstraZeneca. At Takeda, she was Global Vice President of Clinical Development and Clinical Science Head for Gastroenterology. So she is highly knowledgeable, highly credible, and is one of these amazing people that is incredibly scientifically literate, but also understands commercial activities and how the world of business works. So in this podcast, we talked about her experience at a company called Therapeutics, which is one of the early microbiome pioneers. We also touched on the work that she's doing in her current role at the Beyond Celiac charity. And Dr. Silberg has also supported the journey in Prebiotics as one of our key medical advisors. So we talked a little bit about that as well. I think there'll be something in this episode for everybody. She gave some great insights and knowledge into what she recommends to people when they ask her how to improve gut health in the microbiome. And you'll get a lot of insights into how the world of drug development works as well, which I think sometimes can be quite opaque. So with that, I wanted to thank you, the listeners, for continuing to listen to the podcast. We can see our metrics. And despite not having logged a post for a while, you're still listening and you're still engaging, and our followers continue to go up. And that gives me a lot of energy to keep doing this in my spare time, which becomes increasingly little. As always, if you've got any questions, please reach out to the podcast email, reach out on LinkedIn or some of our socials, and we'll get back to you. With that, be well, trust your gut. Thanks so much.
Thanks for coming on Inside Matters. We've been working together away from the podcast on some exciting projects for quite a while now, and I've absolutely loved having your expertise and experience supporting us at Prebiotics. And I'd like to start by kind of asking how you got into gut health and GI rather than any of the other fields you could have got into when you were training to be an MD.
Well, it's a bit of an interesting story, I guess. So I was first a basic science person. I was getting my PhD in immunology before I decided to go to medical school. So I already had a basic science training. I wanted to do something that I could combine the two after I did medical school. I decided to go in internal medicine. But as I was in it, I looked at what fields really needed research. And I felt that there was a real lack of research in the GI field. So it was something that I thought I could add to the scientific knowledge. And then I also met a mentor who was, I would call him really a Renaissance man at the time. He was doing clinical research and basic research, and you can't do that anymore, but he really was doing everything. And he was a great role model, and he was a gastroenterologist. I thought if I could do just any of the things that he was doing, I would be happy in this field. So I decided to go into GI, and I didn't start, well, I've always been clinical some, but I went into a molecular biology laboratory first. And then that transitioned into clinical research after that.
And at that time, kind of what was known about how GI disease developed, what was the current state of thinking? Were patients well served or underserved? How was the landscape back then?
Yeah, and not great. They knew a lot about the physiology. So, but not even, we take it for granted that we understand the hydrogen, potassium, ATP pumps, that makes acid in the stomach. They really didn't understand that. They knew about acid. They didn't know about Helicobacter pylori. There was a lot that we didn't know. A lot of it, we thought that stress caused ulcers.
Right.
Yeah, so all that stuff transitioned while I was in the field or right before I got in the field, is that we started to really understand a lot more about why people had GI diseases. But, inflammatory bowel disease is another one. There were no medicines, pretty much just steroids for that.
Wow.
And so, we've made incredible progress, and forget the microbiome. Nobody was
Yeah.
thinking about
I was just about to ask.
nobody was thinking well, even Barry Marshall, he had to prove it on himself that
Right.
Helicobacter was an
Right.
issue. And I give a course at our local college for adults on called Making Sense of Your Gut. And one of the things I do is I talk about that whole process that Barry Marshall went through by giving himself the bacteria to show that it caused disease. And they're like, really? Someone would do
Yeah.
that? But, at the time, you had to prove Koch's postulates, someone
Yeah.
had to prove it. And, I think now we take a lot for granted that we know, but we really didn't know all of that then.
Just on the Barry Marshall story then. So my understanding is that, and we've talked about it, I think once before in this podcast, but the thesis was you get ulcers because you're stressed and people would go to the doctor and they'd say, take a holiday, relax a little bit, that kind of thing. But, he had this inclination that maybe there was something else going on with regards to H. pylori, and he did like a clinical trial on himself, basically.
Mhm.
So what was the story there? He took it or
Well, so first he worked with the pathologist who they discovered that there was this bacteria and they discovered what it was. And then he took a cocktail pretty much that had Helicobacter pylori in it and drank it and showed that he got an ulcer from it. Uh,
wow.
yeah, it not only was what actually happened to a lot of people is the holiday actually made them worse often. It was
Oh,
because the two things have nothing to do with each other, but, you
and then
probably
you drink a bit more
more
and
exactly. So but when I started out, one of the, biggest or the most reason for an emergency call was someone having a bleeding ulcer. And, we it's really
interesting.
hard to stop a bleeding ulcer with,
Mm.
just, clips or whatever. We really couldn't do it. Everything changed when, Helicobacter pylori was found to be the cause.
Mm.
Of course, NSAIDs can be the cause too now, but,
Yeah.
that was, so much has changed in terms of emergencies.
So before we go any further in your career, I'd love to learn a little bit more about this understanding your gut course that you do for people who don't know much about it, because that's kind of what we're trying to do here on the podcast. So how do you structure it then for the college students who may know nothing other than they might have heard about a probiotic or seen something on social media? How do you structure your talk or your course? Um,
Yeah, so I have it's sort of 4 parts. So the first part is just what are the GI organs and what do they do? How do they work? Starting from the mouth all the way down, top to bottom, as one would say. And
yeah.
then go through each of the organs. How does the pancreas work? What does it do? What does the liver do? Esophagus. Um, how do you absorb materials? Does it get absorbed? All of that. That's one section. Then the next 3 lectures are about diseases of each of the organs, just so that they get an
Mm.
overview of what's going on. Then the 5th lecture is on the it's kind of the microbiome, but it's also what's a prebiotic, what's a probiotic,
Okay.
to try to explain that and why is it important to have prebiotics.
Uh-huh.
To me, prebiotics are much more important than probiotics. You want to diversify your own microbiome as opposed to trying to, because we don't always know, are you getting the right ones? It's much better to feed and diversify what you can have. And then the last one is uh, how to improve your gut health. And then talk about diversifying the plant-based materials
Mm-hmm.
that you eat and different kinds of diets. Why is a Mediterranean diet better than a typical American diet?
Right.
And
That sounds
how
awesome.
yeah. So that's kind of how the course was. What's so funny is I live in a very small town of only about 5,000 people, and about 50 people took the course. I'm going to be repeating it again in September. I run into people on the sidewalk in my little town constantly saying, I took your class. I'm doing this. I'm trying this. I'm eating
Wow.
vegetables and fruits. And so,
That's great.
yeah, it's really,
Kudos to you. Yeah. And are they really? Did they really get into it? Because I spoke to someone else recently who said they're going. The university students are going crazy for gut health at the moment. They want to just learn more and more and more. So, what kind of reaction have you had over in America?
so this is adults. So these aren't college students.
Ah, okay,
This is
interesting.
it's called, I live in a town that has Washington College. So it's the Washington College Adult Learning. So,
Okay,
it's all adults, and they are so interested. They asked me to do
yeah,
it again. And then
yeah,
in April, I'm going to do a new course, and that one's going to just be on gut health and diet.
that sounds lovely. That sounds great. Yeah, keep me posted with that because there's probably some learnings that we can get from all the learnings you're going to get creating the course materials because this is what it's all about on the podcast. This is about
Well,
helping
I've been
people learn.
listening to your podcast, and I'm getting a lot of ideas from that too.
Oh, great.
But one thing that I make very clear, and I think it's really important, is that you should not eat to live. You should live to eat. Meaning, don't make your diet so restrictive and so unpleasant for you that all you're doing is eating so that you can live. You need to be able to live to eat, and eating should be something that should be
Love that.
pleasurable.
I love that. Yeah. That
And I
sounds
think
very
you can,
Mediterranean as well.
yeah, and you could absolutely do that. I think it's just selecting the right things and having,
Mm,
the small portion be the meats and,
yeah.
things like that, and the rest be the plant-based and, trying to really enhance that.
Yeah. So you're touching now on maybe what the best kind of diet is for the microbiome. So maybe we can just zoom in on that a little bit, like based on all of the research you've done and your career to date and preparing for the courses, have you identified like, an optimal approach for diet? You've talked about living to eat rather than eating to live. Anything else? Talked about the
Well,
plants, prebiotics.
yeah, so what I try to tell people is that you can't, it's like feeding anything. If you feed the same thing over and over again, you're really not going to diversify. What you have to do is expand that. Think about other things you can add to your diet. If you're only eating blueberries, add blackberries and strawberries and
Hmm.
raspberries and things because your microbiota need a variety of things to be healthy and expand. So that's one thing. The other thing is fiber. The American diet, and maybe also probably the UK diet too, is very low in
Definitely.
fiber. And try, like, what things can you do to increase fiber? So chia seeds, I mean, things that you can just add in, or, and the other thing that's really important that I teach them is read labels. And a lot of times people are, well, forget the ultra processed. That's the first thing I tell them to take out of their diet. But
Yep.
look at what is actually in the food. So if you're eating something that has just a lot of sugar, that's not good. You need complex carbohydrates because you want your bacteria to be digesting something, not just giving them sugar. Because, it's also the balance between what do you want, what good and bad bacteria do you want? And
Mhm.
also, but it's not like eliminate it totally, but maybe use honey or maple syrup instead of, I mean, and they're all sugar. And that's
Yep.
another thing I tell them too is that, if you break down lactose, it just goes into two different sugars. It's not like a major,
Right.
you're not doing much. But, and also different sugar. Sugar is sugar. Glucose is
Mhm.
glucose. It doesn't matter where you're getting it.
Where it's come from. Yeah.
But sometimes it does add other things. So when we eat honey, you're not just getting the sugar from the honey, there's other things honey has in it. So it's the refining that's the issue a lot of
So
times.
it's like eat whole foods, create diversity, avoid sweeteners if you can, that kind of thing. And it seems to work.
Yeah, I'm really against artificial sweeteners.
Hmm.
I just
Yeah,
don't
me
think
too.
we know.
Mhm.
And I just really worry about what we're gonna learn in 10 years.
Yeah.
To
Yep.
me, it's just
Yep.
better. So I'll use for a sweetener, I'll use like agave sometimes instead,
Mhm.
or something that's more plant-based. It's not an artificial sweetener, it's just a different type of
Right.
sweetener.
Yeah, yeah.
I don't know,
This is
does that
great.
really make a difference? I'm not sure. Makes me feel better, I guess.
Yeah, absolutely. And maybe it's just that little things stack and compound. And, if you're doing something every day, you're going to get benefits. And if it's a bad thing like a sweetener and you're having it 3 times a day, maybe you get a stacked effect towards the negative. So I think what you're saying makes so much sense. Start with the basics and
Mhm.
still enjoy yourself from time to time. I think that's super important.
The other thing
Um,
I get is a lot of people ask me about gluten-free. So, I you probably know this, but one of my other jobs is I'm the chief
yeah.
scientific officer of a patient advocacy group called Beyond Celiac, and so it's something that I'm very interested in. I do not have celiac disease. But when people say, "Oh, I think it's healthier to go on a gluten-free diet if they don't have celiac disease or a gluten sensitivity," I say, "No, it's not. It's actually less
Mm,
healthy because the way that our wheat is processed, we actually put in uh, some of the nutrients that you actually need. And if you start eating gluten free, it's often very, very processed, and you're also getting a lot of rice. You can maybe not, but a lot of things are rice. Rice has a high arsenic um, uh, content in it. So over time, we don't know
mm,
how that might accumulate.
mm.
Plus, to make it taste better, a lot of gluten-free products have a lot of added stuff like tapioca
Interesting.
and sugars, and so
Interesting.
it's people who have celiac disease who really have to be on a gluten-free diet actually gain weight usually.
Oh, really?
Yes, yes,
Huh.
and it's
That's really
also
interesting.
very low in fiber.
Yep. I've read that before.
So I tell people, unless you absolutely have to, don't go
Uh,
on a gluten-free diet. It's not a healthier diet.
mm. We've not talked much on this podcast about celiac and gluten-free diets, likewise gluten sensitivity. So it'd be great to learn a little bit more about that because a lot of people, I think, say I'm sensitive to gluten, but is there like a medical definition for that versus celiac versus not at all
Well,
based on your experience?
yeah, I mean, celiac disease is an autoimmune disease, and it's actually quite a severe disease. There was just a paper published talking about the increase in GI cancers related to celiac disease. And generally, it's when you're not really careful, or I would say it's more late diagnosis of celiac disease. So you've been eating a lot of gluten containing products. What happens is you get constant inflammation. There's a huge cytokine burst for people who have celiac disease. And so your immune system is being activated. And that's just not good for your intestinal tract. And that's primarily where the damage is. However, we also know that there's a lot of neurologic damage that occurs too.
Really?
Yeah, there's a lot of
Wow.
studies actually in Sheffield, UK.
Huh.
They're doing a lot
Okay.
of studies on trying to understand the neurologic complications of not just celiac disease, but gluten sensitivities. So you can get ataxia, um, not just for celiac, but some gluten sensitivities that can be very, very
Wow.
severe.
Wow.
Depression, anxiety, all of these things, brain fog, fatigue, all associated with both celiac disease and some gluten sensitivities. But for celiac disease itself, that is an autoimmune disease. And I mean, it's as severe as any autoimmune disease, lupus, and I mean, really can have a major effect on your body. Gluten sensitivities is more of um, it's a wide term. There are absolutely people who have gluten sensitivity who do not test positive for celiac disease. And they can have paresthesias, different other manifestations. I've heard of people having a cough associated with gluten. So there's a lot of things that you can have. And I think this is where, eliminating and then putting things back is really helpful. We now are understanding a lot more about these other more allergic type diseases like eosinophilic esophagitis, where we have an allergy pretty much to multiple different foods, or could be an allergen, an external allergen. But now what we're understanding is that you can probably eliminate a couple of things
Mm.
and see if that works, as opposed to
Yep.
eliminating like a huge amount of things.
Makes sense. On the gluten sensitivity versus celiac, do we understand why someone might be more on the kind of sensitive end of the spectrum versus someone who might be on the celiac end? Is it genetic? Is it microbiome? Is there something that we've identified that determines the sort of severity and spectrum that you were describing there?
So one might be more of an allergy. So that's a
Mm.
different mediation. That's more of a like an IgE type mediation, where uh, an autoimmune disease, in celiac disease, there's both IgA and IgG. So it's the immune response. But celiac disease has a specific HLA characteristic, so a genetic
Yes.
predisposition. But not, I mean, like 40% of people have the gene, but only 2% of the whole population gets celiac disease. So it's a predisposition. And it's actually those same people also have a predisposition to type 1 diabetes, uh, autoimmune thyroid disease, because that's how HLA, that's what HLA is. It's the ability of your immune response to react to certain things. Now, what's interesting, you asked about the microbiome. We're
Um,
actually funding some research to look to see, are certain bacteria more prone to digesting gluten in a way that makes them more immunoreactive. And we think that
wow.
it's the case.
Really?
Yes, that there
Wow.
is a a certain type of digestion. We're trying to
So
find out what the bacterias are that might be doing that,
wow.
but that's a potential therapy.
Absolutely. And also it might explain why if, only 2% of the population, sorry, 2% of the population carry the sort of genetic predisposition, but a
No,
smaller number.
40% carry the predisposition, but only 2% get the disease.
Oh, okay. So do you think the 38% delta could be driven maybe by the microbes making the gluten more immunogenic? Because that would be
That's possible.
staggering.
Yeah. The other thing is, we know that people who have used antibiotics frequently in their early years have a higher
Interesting.
likelihood of getting celiac disease.
Okay. So that's another aspect then to the microbiome story, but
Hmm.
that's really interesting.
Yeah.
Wow, okay. So when will you get a readout from some of that research to try and, say one way or another?
Well, I mean, they're working on it, and it's an area that we're interested in because,
Yeah.
I think that people with celiac disease in general and the people in this field are really interested in understanding if there is a potential treatment.
Yeah. And just so I understand, at the moment, there isn't a treatment, but it's removal of gluten from diet? Yeah,
That's the treatment.
yeah.
Right.
And is there like a pipeline of interesting drugs coming through, or is it a
Oh,
tough area?
yeah. Oh,
Yeah,
well, I would say almost every large um, pharma company is looking at celiac disease as a potential area for treatment.
interesting.
Yeah,
And then
a
you
lot of the
contrast that
big
to
ones.
IBS where there's very few. I wonder why. It's
Yeah,
interesting.
yeah. Well, one of the other things is, and this, it happened just coincidentally, but I think it's interesting, is there was a patient who had celiac disease, but also had C. difficile, and was treated with an intestinal microbial transplant, and their celiac disease got better.
Wow. So maybe some proof of principle there, although the critics would say very small sample
Of
size.
course.
Yeah, of course. But that's interesting.
Yeah, yeah.
That's
I mean, I think
interesting.
it's really fascinating. The
Mm.
potential there is that if you can figure out which of the microbiota is causing this issue.
then you
Yeah,
might be able to eliminate them or outcompete
right,
them or so on.
exactly.
It's
Yeah. Mm,
fascinating.
yeah.
Awesome. Now we've done a lovely deep dive into celiac, which is great because you're actively involved there. I just like to take you back a little bit.
Sure,
So
good.
yeah, I'm always interested in understanding from people, when and where and how did they first hear the word microbiome, and what did you think, and how did that then lead you to what you ended up doing later, which was working in that specific space?
Oh, boy. That goes back a long time. So, boy, I would say so I don't know if you know Gary Wu from the University of
I
Pennsylvania?
don't
Oh, okay.
know.
So, he's one of the professors that when I was at Penn, he worked there as well, and he got really interested in, the microbiome. He was one of the first people that I knew who was working on it. And then he got very involved in the American Gastroenterological Association, put a task force together, and he was involved with that. So he was probably the first person that I had really heard about working on this. And um, he came to me with a proposal when I don't remember if I think I was working at Shire at the time. And it just made sense to me. And after
Mm,
that, I was like convinced that there was so much there that you could do. I think I'm also very open to
mm.
possibilities. And
Aha.
though my senior leadership was not as interested, I said, I think this is going to be like the future. I
In
really,
when
and I
and you're
still
in big
believe.
pharma company. Yeah.
Yeah, in big pharma, well, the other thing is they couldn't see it as a drug. That's the problem, is that you have to imagine and
Mm,
you have to understand that not everything. I'm sure people, when we were doing monoclonal antibodies for treatments, people said, well, how are we going to make this work for a lot of people? You have to imagine that that there's going to be a solution.
mhm, mhm. And, just take us back then. So at Shire, you were focused on GI drug development,
Yes.
and you developed a belief, but how did you end up working on microbiome drugs? 'Cause I think there's a bit of a jump between first hearing about the term microbiome and then working on the drugs.
Well, then Takeda bought Shire, and I was the
Ah,
head of clinical development for internal medicine at Shire. And then I became the head of clinical development for um, GI at Takeda. And they had an interest in looking
okay.
at the microbiome.
Got you.
And so that's then how I got more involved in it. And after I left Takeda, I worked for Finch, who
Mhm.
was trying to develop drugs for using the microbiota.
And could you maybe give the listeners then some perspective on what might be some different therapeutic approaches to the microbiota based on what you've seen over the years, maybe single strain, consortia, FMT, and give some impression on, you know, what might work the best,
Yeah.
perhaps?
So my philosophy, at least, is that you need to have something that's broader unless you know specifically what you're treating. So I think that C. difficile was sort of the lowest hanging fruit
Uh,
for drug development. All you really needed to do was compete with the C. difficile. It didn't really matter so much, I don't think, what the components were, as much as you just needed to populate the GI tract with something that was going to compete. That worked. The problem is when you get to other diseases and you're not really sure the mechanism, you can't know that 3 or 4 or 6 bacteria are gonna be the only ones necessary. And the microbiota is complex. The whole microbiome is very complex. It's not just the bacteria, and it's not just the anaerobes and the aerobes. We don't know how much does, uh, fungal and how much does viruses, there's so much more. So what I would say is you probably wanna go, broader from a healthy individual to
mm,
repopulate unless you know what the mechanism is, than to
mhm,
try to pick out 5 or 6 strains because you don't know if that's all that's needed. For me, it's the same thing with food, is that you can't just choose one thing. it takes a multitude of things, and that's why you eat
Right.
whole foods and not just one
Yep.
part of a food.
Yeah. So how does that fit in then to the probiotics industry? Have you got any views then on the probiotics?
I I hope it's not blasphemous, but I'm not a big probiotic fan.
It's not blasphemous at all.
I just think that
Yeah.
we again, it's the
Yeah.
same thing. Well, also, most probiotics, unless you take them every single day, they don't, engraft. They
Yeah.
just go right through. I mean, they might do
Yeah.
something when you have it, when you're eating it. Now, not to say I'm not against fermented things. I think yogurt
Right.
is great to eat, um, but not because of the probiotics, but more because of the fermentation process that goes on.
Exactly. So you're getting the benefit of the fermentation process, the food, and then as a byproduct, there's also potentially some live microorganisms in there
True.
too. So it sounds like you're a big proponent then maybe of food as medicine rather than a specific strain of
Right.
bacteria. Is that is
Yeah.
that fair?
Yeah, I think so. I mean, I'm not a big kombucha fan, but I don't have anything against it. I think it's probably good. Um, just to me, just the taste is not great. And like I
Yeah.
was given yeast ice cream, not a fan.
Oh, that seems well, I'll tell you, it's very different from what we have here in Scotland where we have deep fried ice cream. So that gives you some deep fried everything. Everything's deep fried over here.
Sounds like
So maybe
Wisconsin. Yeah, we have. There's a state that like everything they fry in butter.
everything's
Yeah.
deep fried in batter. So yeah, so going back, so maybe it'd be good to learn more about what are some of the challenges associated with microbiome drug development specifically. And what's unique to maybe developing a full spectrum drug versus a small molecule or monoclonal antibody? And what's hard about that? Because we've had only 2 approvals and the field's been kind of hot for 10 or 15 years now, I would say.
I think it's probably consistency over batches
Uh,
and over time is going to be important. And the reason I say that is, when you have donors, every every person's microbiota is going to be different, or the whole
mm,
microbiome is going to be different. Every person. Two healthy people, they're not going to have exactly the same things. They might have proportions similar, but it's going to be very different, and it's very much related to what you eat. So it can
yeah.
change over time as well. So I think that that's going to be one of the more difficult things for the future is consistency, and does it work, you know,
Um,
10 years from now, the same thing that you started with, is it really the same thing you end up with? Because our diets are constantly changing as well.
mhm.
So that
Maybe I could press you a bit on would a FMT or an IMT produced in the UK or America, do you think that would be effective in Japan?
so I know that's a concern actually for the US, for Europe,
Mhm.
is whether or not it's
Right,
translatable.
right,
Um, I don't know. I don't think we've have enough studies. Is there the certain secret mix that you need?
right.
I don't know. I mean, I know that there are people who are trying to do like an external intestine, kind of for an internal uh,
Yep. Robotic
milieu.
gut,
Right, exactly.
robo gut.
Yes, or probably to make it more consistent. But again, I think you have to make sure then, do you have all the components? You don't want to
Right.
end up not with something that you really do need. So, but this is where the biggest challenge, I believe, will be is this consistency
Yeah.
across, batches and time, and making sure that it's always gonna be working.
Mhm, mhm. And when you were working directly at the coalface with some of these microbiome therapies, what were you most excited about when you were developing these drugs? Was it the C. diff story, or did you have this broader vision of maybe IBD, IBS, celiac?
I think that I don't know, there just seems to be a really wide potential. because the gut and the brain are so connected that IBS is actually a perfect place for that connection,
Yeah.
but also mental health
Mm.
in general, even with people who don't have IBS. One of the things we were looking at Finch, one of the programs I was looking at, it was autism. And the question was, are you treating just the constipation? Or is it when someone who has autism has better gut health? Does that influence their general health and mental state?
That's interesting.
Unfortunately, we didn't really get a
Yep.
chance to do enough to be able to do that. But there are people who are still working on that area and trying to understand that.
And what's the story there is, well, the gut and the brain might be connected, and if the gut's not working properly, then it may have an impact on the brain. Is that the sort of general thesis?
Yeah, partly. It's also the metabolites that,
Yeah.
so if you look at children who had autism, some of the metabolic features were potentially from bacterial metabolism, that if you could change that, and it wasn't to cure autism, it was more about making children, especially children with autism, have better lives because they may be able to think clearer, not have anxiety. So that was really the whole point.
And was Finch looking at the kind of GI symptoms, or were they looking at cognitive function as well in the clinical studies that it was planning?
Both. So GI function and cognitive. There was a
Wow.
lot of cognitive testing that was going to happen as well.
Wow. Wow.
Yeah.
Well, it's a shame that didn't move forwards, but maybe at some point someone will, and see if they can, they can show that link
Yeah.
between the gut and the brain, because it's a big unmet medical need, I think, isn't it?
Right. But
Um,
I could see this in other mental health disorders too, anxiety and depression.
mm, absolutely. We had some evidence of that in our liver cirrhosis study, where the anxiety and depression scores were improving in a dose-dependent manner. And some of the key opinion leaders that were looking at that data were saying, well, actually, depression is very common in cirrhosis patients. You can't help but wonder if maybe these nasty gram-negative bugs that are maybe overabundant were contributing to some low levels of inflammation, which systemically find their way through the gut to the brain and maybe have an impact.
Yeah.
Be great to see that in a bigger study.
I would think too that autoimmune diseases, even if you're not curing the
Mm,
disease itself, you might be able to tamp down some of the immune response,
mm,
because we do know that the microbiota do contribute to inflammation.
mhm, mhm. I think that's absolutely a route forward as well. Although one of the things you've been very clear on when you've been supporting our development is focus.
Focus. Well, look, I think it's just a general thing for small biotech
Yeah,
companies. Is it you
yeah,
have to get the first win,
yeah,
and then you can start expanding. But
yeah.
without that first one, I would say if you look at any company and how they started with one win and then
Mhm.
were able to expand to other things that they were interested in.
Yeah, I think that's completely fair. You wanna do it all, and you wanna do all this exciting stuff, but you've just gotta lock down on one thing, and
Right. And
and I
do
think
that one right.
get great data.
Mhm.
That's the currency, isn't it, of the realm?
Oh.
Um, so how did you transition from being a doctor into developing drugs? What got you excited there?
Um, can I say that I hated my job at
Yeah, of
first?
course.
Well, no, I didn't hate it. So,
It was less exciting than the other opportunity.
yeah,
Yeah, I can
so
relate.
so I was at the University of Pennsylvania, and I was a basic science researcher. And I had, I think at the time, maybe 4 NIH grants. I had like 2 R01s. I was running a center. I wasn't running the center. I was running the immunohistochemistry type thing of a center. And I thought, okay, I've got 4 grants. I'm really well funded, and I do not like what I'm doing. I have to find something else to do. I said, if I've had all this money and I'm not happy, then this is not where I need to be. So I
Fair.
started looking for other alternatives, and I talked to a lot of people. And the people who seemed the happiest were the people who went to pharma from academia. My academic colleagues thought I was crazy and how it was
Oh,
going
yeah.
to be terrible and how I wasn't going to be able to talk to anybody. And
Yeah, you're going to the dark side.
it was awful, but I made that transition. It was the best move that I ever made. I
Oh,
was
wow.
so happy. I love pharma. I was Not only did I not feel stifled, I met people that I could never have had contact with that were leaders in the field that would never have talked to me before.
There you go. So there's now going to be a mass exodus of NHS junior doctors as a
Sorry.
result of that statement. So good luck to the NHS.
There's you know what? There's a place for everyone. And I mean, a lot
Yeah.
of my colleagues stayed at Penn and are still doing
Mm.
research at Penn. It just
Yep.
wasn't for me. And I love what I ended up doing. So
Yeah, and you can feel the energy in your voice, and I mean, I'm the same. I spent much less time in the NHS, I think, than you spent in the hospital in the US, but it's quite exhilarating developing a new medicine. The challenge associated with it, it's just so intellectually stimulating, and you've all these stakeholders and so many big decisions to make. The data comes in. It's great. So what have been some of the highlights then, the most interesting drugs you've worked on, key learnings, progressing to, senior leader in drug development roles? What do people get wrong about drug development? How do you get it right?
So one of the biggest things people get wrong about is stopping a project or a compound and not just keep on trying it again and again and again. And I found this in industry all the time is they have something, so they have to figure out where to use it.
Mm.
And I was never like that. I had a drug, actually it was an IBS drug. The placebo did better than the active because
Uh-oh.
because there were so many side effects from the active that people were
Right.
feeling bad. I said, this is crazy, or liver. I was because I was a gastroenterologist, I headed at AstraZeneca, I headed their liver, their kind of DILI, their drug-induced liver injury panel. And a lot of times teams would just ignore the liver signals. And my general philosophy too is you can't be worse than than the disease you're treating. So you can't give somebody something just because it works if it's going to cause damage to something else. And that's another one is submitting an NDA for something that has a serious adverse event. Nobody's going to approve it. And you shouldn't get to that point of submitting an NDA where someone has to tell you, no, it's not safe.
Mm-hmm.
You have to
That
really
makes complete sense.
So those are the things that I learned. As far as successes, it didn't happen until after I left, but I did bring in and then develop a drug for eosinophilic esophagitis. It's a budesonide slurry. And that was exciting more because there wasn't anything to treat that at the time. And people were mixing, they would take packets of Splenda. I don't know what's
Oh, yeah.
they would
Splenda is
it's an
it's a
artificial
sweetener. It's a
right.
sweetener. Yeah.
They would
Yeah,
take
yeah.
that with their asthma medication, the budesonide and an asthma medication, and mix them together
Oh, wow.
and then take them. No clinical trials to say
Wow.
what would happen, but doctors were doing it because they didn't have a choice. Well, we developed a formulation for that, and it did eventually got approved. That was
That must have been awesome getting the FDA registration.
Yeah, that was fun. It happened after I left, but it's still something that I worked on that I was really proud of.
Could you give us a sense? Some of the listeners might see something in the news that says, breakthrough treatment for disease X, and it's an animal model. Maybe you could give some sense on, why it takes so long for some of these promising, you know, preclinical results to become an approved drug. We talk about the journey,
Yeah.
high level, what's required, because a lot of people won't know what's actually required.
Yeah. Although I don't know if you're reading the news lately, the FDA is now trying to do this whole AI thing and not do animal studies, but that's a whole nother story.
Yeah,
All
I
right.
think you might have some strong views about that just
I-
based on previous conversations.
Well, let me tell you about the drug development story. First of all, all right, so first you have a concept. The concept then has to be put into whether it's a model for a cell model, an animal model, it doesn't really matter. You have to have a hypothesis of how something's going to work, and you need to then test it. And it's better generally, if you can, to test it not in a human, but in either an animal model or a cell model. I mean, we now have organoids, so we make intestines, say, in cell culture, and then you can see what's happening. So you have to do that because you have to prove that this mechanism might work in some way. That can take a few years to just do that part of it, to really test it and to understand it. So, you're talking about the very initial stats. Once you get something that looks like it might work and you want to put it into humans, you generally have to do healthy human studies. So you have to get the compound in a safe, well, we don't know it's safe until we try it, but what I mean is you have to put it into something that will not be harmful to a healthy individual. And then you usually test this in healthy individuals. And sometimes what you're testing is not just the safety, but also what we call the pharmacokinetics. So how does it actually get metabolized in the body? Excuse me, where does it go? How much do you need? What are the levels? And you have to really start at a low level, and you usually do a single dose. We call them a SAD and MAD study. So you do a single ascending dose study. You start very low, then you go higher and higher and higher, just to make sure that it's tolerated, and you can look at the pharmacokinetics and how much that person's actually being exposed. You generally get to a certain dose where you feel comfortable that that might be the right dose. Then you do a multiple ascending dose study, and that is you give the dose. You have to figure out, is it once daily, twice daily, 3 times daily, whatever it is. Then you try that in usually healthy individuals, and then you do an increase in that too. And you try to get to a level where you're not hurting somebody, but they can't tolerate it anymore, so you know what the maximum dose is. Once you've done that, this is kind of traditional drug development. Then you can start looking at how does this act in someone who actually has the disease. And we usually do what are called these are all phase 1 studies. You do that phase 1 study, and now people who have the disease, you look at the same kind of things. Do they metabolize it in the same way? What's their top dose? And you can start looking at whether or not it has any effect on the disease in question. But sometimes you're just doing dose finding, and you're not doing it long enough to really know how it's going to affect the disease. Once you have that and you have an idea of what the dose is going to be, you go into what's called phase 2. In phase 2, you're more interested now in looking to see what we call the pharmacodynamics now, not just the kinetics, not just the metabolism. Now you want to know, how does it actually affect the disease itself, and how much do you need, and what kind of intervals do you need. So that's usually the phase 2. They're usually shorter studies, but very concentrated on safety, tolerability, and some efficacy. Once you figure that out, then you can go, sometimes we do 2A and then 2B. So 2B might be in a more extended study. And then you can go to phase 3, which is usually, we call those registrational studies, where you now know usually what the doses you want. And now you're going to do longer studies to see what the effects are. So, you're talking, depending on
A long time.
Yeah, I mean,
So much work.
it's like
Yeah.
11 years for, and not only that, each compound that goes into humans, there is only about 1% or maybe even less get to the point of even getting to phase
Wow.
3. So it's a lot of attrition. So if you're an investor,
Yeah.
just know that nothing is it's not a fair bet. But if we don't have these things, you can never get to the next step. We learn every time something fails, we learn and we build on that. And that's how you do research.
So basically, there's a huge amount of work and there's attrition at each step. And what we also didn't talk about were all the manufacturing challenges associated with scale up. And there's a whole another sort of domain there where it takes a lot of time and effort and energy and money and innovation. And that's why drugs are expensive, right? A lot of failure.
I was working on a drug and we could not figure out how to make a pediatric formulation for it because it had this really complex capsule and the kids needed a liquid form and just could not figure out how to do that. And it's really important. Every drug that should be available to children has to be developed in children. And that's it's a good rule.
It's a good thing. It's a good rule.
It's hard sometimes to do that.
Absolutely. So maybe that gives some clarity. That was a really awesome explanation on how you go from an initial piece of evidence right the way through to sort of definitive evidence. And I think that will help the listener understand why it's so difficult. Could I just, maybe for a bit of fun, just press you a little bit on the FDA's AI animal mod? I just might be. I know you've got some strong views because we discussed some similar things off the podcast. It'd be great to hear, what's going on in the US right now. It's a bit crazy.
It is
from the outside looking in. Yeah.
I hope I don't get in trouble for saying any of these things. Someone will knock on my door. My concern is AI can be very powerful.
Yeah.
But it's only as good as the information that it can access. And there's a certain amount of human filtering that's necessary to know what's good and what's not good information.
It's true.
And oftentimes the way that AI works is it just uses everything available, which means, anything that anybody puts, it doesn't have to be from a reputable source. So someone has to be looking at it and making sure that that's true. From my understanding of what they're trying to do at the FDA is to do initial reviews of documents that are being submitted for filings to cut down on the amount of work people have to do. I don't think that's a bad thing, but I do think you need oversight. And it's true. I mean, when we used to have paper filings, you would have a semi truck, a huge truck filled with millions of pages of documents. Now it's all electronic, but you're talking about 500,000 pages of material that people have to read through. Submissions are big. You're talking, each of the parts of a submission, from manufacturing to the clinical trials to the safety data, all of that has to be reviewed. If there's a way to do that faster, that's great, but you need to have good systems in place to do that. And human oversight who's going to catch that, no, they use something that is actually not valid.
Right. So it's an enabler rather than a replacer.
Yes.
Yeah.
Yeah.
And I want to ask you about your views on the current administration, because that's separate. That's off air. So we've talked a lot about microbiome, what's good for gut health, drug development, covered a really, really broad base, actually, and it's been an amazing conversation. What I'm trying to do with guests now is finish up with a quick fire round of questions. So I wanted to just do that with you and see how that goes. So the first one I wanted to ask you is, has the microbiome bubble burst?
I think we're in its infancy. I'm just
Great.
I mean, I'm really hoping that this is going to continue and expand.
Mm.
That's where I'd like it to go, at least.
Yeah, because there's been a little bit of maybe too much hype at some points, and we haven't quite delivered in some areas. But I'm really appreciating your optimism. I would say the same for
Okay.
sure. So that's great. What are you most excited about right now in terms of microbiome research?
It's really anything having to do with gut brain to me is really important. So
Mhm.
I would say that area. Well, it's two things. It's not just the gut brain, it's also the inflammation. I think it's those kind of things that really make me interested and think that there's a real need to move forward. And
That's
again,
great.
it's not just about, and I know that's what you're doing is intestinal microbial transplant, but I think it's
Yeah.
also about understanding prebiotics as well, to just getting healthier diets to help with some of these things. I don't know that we're going to cure everything, but I think we can make people feel better.
Mm. Yeah, we I guess we're kind of in the gut-brain axis with IBS, aren't we? In
Absolutely.
Therapeutic. Yeah. Yeah. No, that makes sense. And then when you do your lectures to anyone who's interested in gut health or the microbiome, is there any one particular fact that you say that gets people excited? Or, when you talk about the microbiome, is there any particular aspect about it that makes you think, wow, this is really amazing?
So I give them some examples of things that we are learning. And not all of them are necessarily going into clinical trials, but some are. Things related to cancer, for
Mm.
example. So, like melanoma work. I was really fascinated by, and I don't, I would say they're more anecdotal at this point, but where you get a super responder and you
Yeah.
then transplant their microbiota to someone who's not a responder and that person responds. I mean, that kind of data is just really interesting and impressive. And
Mhm.
I give those kind of examples to teach about the possibility of the future.
Right. Yeah, I mean, imagine being able to potentiate an immunotherapy through an IMT or an F. That would be epic. Great. Well, that was everything I wanted to cover. I think, looking at the agenda, we got through everything and more. So I wanted to extend my thanks to you. And yeah, I really, really enjoyed this conversation. And, separate to the podcast, wanted to thank you for all of your expertise and guidance as we've gone through our IBS trials. And I hope you've been enjoying it as well, working with our small entrepreneurial dynamic team. And it sounds like you're also a listener to the podcast as well now, which is great. So
Well,
a listener and a guest.
it's not only been a pleasure talking to you today, but it's really been a pleasure working with your group. And
Thank you.
I think you really have some very interesting data. I'm
Yeah.
very excited about the future.
Yeah. Thank you. Thank you so much.